Protein. We see it everywhere in our daily lives as they are responsible for the proper functioning of a living organism. We see it in our food, in plants, even when we look at ourselves in the mirror, as we are made of protein. They're in our hair, bones, muscles, enzymes, everything. But if we're made from protein, how is protein made? This is done through a special process called polypeptide synthesis. I know, mouthful, right? A protein is a 3D molecule made up of one or more polypeptides. It starts from an amino acid, which then connects into a polypeptide, which is a chain of amino acids, and then transforms into a protein. The process of polypeptide synthesis occurs in a normal DNA molecule found in the nucleus. Firstly, a copying process called transcription will start, where a section of the molecule will unzip to reveal the genetic guidance to create a protein molecule. The enzyme that separates the specific DNA strand is known as RNA polymerase. Side note, DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. Once it has been separated, a single strand on RNA forms and transcribes the genetic information from the DNA molecule. This RNA strand then turns into messenger RNA, or simply known as mRNA. The RNA only has two of the same bases, which is C and G. This enables the bases to become complementary to each other. A and T also match up, but any time the DNA molecule has an A base, the RNA matches it up with a U instead of a T base. RNA uses uracil instead of thymine. If that explanation seemed a bit confusing to you, here is a more accurate version of what it would look like. The RNA only has two of the same bases, which is C and G. This enables the bases to become complementary to each other. A and T also match up, but any time the DNA molecule has an A base, the RNA matches it up with a U instead of a T base. Okay, got it? Let's move on. mRNA does not copy the whole DNA, as there are start and stop signals. The mRNA will then go on a little adventure and leaves the nucleus, as it is small enough to travel onto a ribosome, which is inside the cytoplasm. As the mRNA strand leaves the nucleus and attaches to a ribosome, the second step of this process begins, which is called translation. The mRNA is read 3 bases at a time, which is typically known as a codon or triplet code. Ribosomes are also made up of RNA, and these are known as ribosomal RNA or rRNA. Once it has attached, the ribosomes work like a translating machine and read the mRNA codes and place the accurate amino acid using tRNA. tRNA stands for transfer RNA, and its mission is to match the proper amino acid to the ribosome. tRNA has anticodons, which are attached on one end. These are complementary to what is found on mRNA. On the other end is an amino acid, and once all have matched up correctly, translation will occur for each 3 sets of bases. This will soon end up as one long chain of amino acids, which is called a polypeptide. As I mentioned before, there are start and stop signals. When the mRNA goes into the ribosome, it looks for a start signal, which is usually AUG. The translation process continues on until a stop signal is found. These are usually UAA, UAG, or UGA. The polypeptide chain can be as long or short as it likes. Just really depends on when the stop signal appears. But the process isn't quite finished yet. The polypeptides must then fold into an unusual but specific shape. There are 4 structures a polypeptide can form into: primary, secondary, tertiary, and quaternary. It is then sent off to the exact location it is needed. And that is the process of polypeptide synthesis and how protein is made. Now, after watching and listening about how polypeptide synthesis works, I'm sure you heard many RNAs being thrown out there, but they all play a key role in the procedure of protein. Our most important are mRNA and tRNA. Here's why. It is crucial for the mRNA and tRNA complete their jobs appropriately and accurately in order to avoid complications that may affect the overall functioning of organisms. If mRNA and tRNA does not complete the coding exactly or not connect to the correct amino acid to the polypeptide, this could lead to genetic diseases such as cystic fibrosis or hemophilia. Remember these key points of polypeptide synthesis: transcription, translation, DNA, RNA, mRNA, tRNA. Thank you for watching. I hope you learned something.